at the genome in a targeted manner at multiple target sites simultaneously. But before we get into this, I understand that a lot of us are going to come from different backgrounds, and not all of us may be super familiar with the biological setting in which we're doing this work. So we've been trying to validate the tool in vivo setting in Drosophila melanogaster neurons. And so we want to just basically have a sort of 10,000-foot view, simple framework of understanding the biological programming of how these neurons come to be. So life as a neuron, in the simplified view, starts as a neural stem cell. You're expressing a variety of transcription factors, think things like deadpan, clump fuss, ascents, that sort of stuff. These neural stem cells, unsurprisingly, can undergo self-renewal and symmetric division, but they can also begin to express prospero asymmetrically and give rise to ganglion mother cells. Prospero is sort of working to, we think, initiate the differentiation process. As this continues, your ganglion mother cell will divide symmetrically to give rise to neurons. And at that point, an isoform of Lila N can come along to sort of cement and maintain this differentiated state. But what's more interesting for us is the sort of things that are happening at a molecular level as this process goes on. So in your neural stem cells, these transcription factors, things like deadpan, are very accessible, and they're not yet laden with repressive marks. As you progress through the differentiation process, though, they become increasingly inaccessible, and they become not exclusively, but overwhelmingly laden, in most cases, with HP1. So, a couple of the aims and objectives that we've set out with this is, of course, first we'd like to develop a toolkit that will allow us to modify these marks as we desire at multiple target loci. And then, provided that our toolkit works, we'd like to try and answer a biological question of whether or not removal of H3K9ME3 and HP1 can facilitate reactivation of some of these neural stem cell transcription factors. Now, before we get into the toolkit too much, there are a couple of core developments that I think are worth mentioning. So we're using Cas12a, and wild-type Cas12a has both nuclease function as well as ribonuclease function. So one of the core developments was from Zetche et al. And so what they discovered is they had a couple of, all good? Yeah. They discovered a couple of single amino acid substitutions, which I've underlined there, where you lose the nuclease activity, but you have retention of its ribonuclease activity. And this will become apparent why it's important later. Nihongaki et al. Were doing some work where they were essentially fragmenting Cas12 into two halves, and they were looking for fragments that would dimerize in the presence of rapamycin. And what's important for us, as far as we're concerned, because we're not going to focus too much on the experiments, is that they found some that are constitutively active, where they readily dimerize, even in the absence of rapamycin. And lastly, Guo et al. Discovered an additional single amino acid substitution that increases the affinity and efficiency of Cas12a for identifying and binding to target sequences. What's really nice about all of these is that you can employ all three at once without compromising the properties conferred by one of the other substitutions or fragments. So this is where you guys are going to have to bear with me because I'm not the most artistically gifted, but I'm going to try and show you guys a nice little animation of how the system works now. So we begin by expressing a guide RNA array. We use U63 promoter. You've got these nice little stem loops that separate your target sequences. Then we can turn on, using Gal40UAS, our split Cas12a fragments. You can fuse to both termini these effectors using flexible linkers. In principle, we'll get into it later, but they dimerize, right? They make their way to the guide RNA, process it, find the homologous sequence, they'll carry out the modifications, and then, if everything works according to plan, you'll get some sort of response from the target gene. All right, so we have a lot of different effectors because we're trying to build quite a large catalog. And at present, we've been focusing overwhelmingly on effectors of proteins that can establish or remove certain modifications to the histones. But we also have ones that should help to recruit transcriptional machinery. We have ones that should, facilitate nucleosome remodeling, among some other miscellaneous things. As far as the nomenclature is concerned, of course, you have, when you split this protein in half, you have a new N-terminus and a new C-terminus. So we write out from the N-terminus to the C-terminus, KDM4B, the N fragment of our CPF1, and then KDM4B. We call these singleplex because all the effectors are the same at each domain. We're going to be focusing on the lysine demethylase 4B. But I think unsurprisingly, you should have this in your mind that all your effector domains need not be the same. So the idea is that maybe we can carry out multiple different epigenetic changes in tandem with one another at our target sites. So maybe we remove a repressive mark and lay down an activating mark. At the same time, we can, like I said, recruit transcriptional machinery and remodel, the position of the nucleosomes, reject nucleosomes if we so desire. There's a couple different ways that we can validate the toolkit. So we have something, sort of like more molecular genetic techniques. So these can be things like, are we actually laying down the mark? Think, chip, seek, cut and run. If you're not doing that, it's not a very useful tool, right? The other thing is, do we see changes to accessibility? So we can go for things like ATAC-seq. And then, assuming you've done all of these things, you want to see if the gene actually responds to the treatment. At the same time, you can use immunohistochemistry and visualization to see, are we getting ectopic expression at protein level of our target genes? And what consequence does this have for the cell? Are you seeing, at least as far as our neurons are concerned, re-entry into cell cycle, interesting morphologies, dedifferentiation? But for our purposes, I'm going to be focusing on the molecular genetics and sort of what's happening at transcript level when we employ these tools. So this is experimental design. Like most fly experiments, it begins with a cross. We turn on the Marvin Gaye music and we let the flies get to work. And I'm also, alongside the system, expressing a GFP. We let these flies grow up, third instar larvae. We dissect the brains. We give them an enzymatic treatment to create a single cell suspension. And then these can be used as input for our techniques. So at this point, I'd like to just talk about very quickly the structure of our guide RNA arrays. So we have one array where it's a very rational, intelligent design where we're targeting these peaks that have been identified through TADA as being very enriched at important regions for expression of these genes. And then we've got a second guide RNA with a sort of less sophisticated approach where we're just throwing it down close to the open reading frame. So one would anticipate that the first guide RNA does better, but that's not the case. We see very little transcriptional changes. But our silly guide RNA2, we're actually getting quite a few upregulated and downregulated genes. And I should say this is relative to a control where we do not use a guide RNA array. So everything else is there, all the lysine demethylase, all the split system, but just no targeting. You might wonder, how do our 10 genes respond? We would only anticipate response from 9 of them because one of you is polyclone group repressed. So you would need something like a UTX to remove its repressive mark. But of the 9, none respond for our first guide RNA array. But our second guide RNA array, we see a very small, modest response from DEDPAN and M-gamma HLH. And when we look at our upregulated genes, we see some interesting stuff. It's not an exhaustive list, of course, just showing you the ones that are sort of most relevant for what we're trying to do. But when we look at our upregulated genes, we see overrepresentation of terms for leading strand elongation, DNA replication, things that you don't think would be important for a cell that's post-mitotic. But we see underrepresentation of terms where you'd think they would be important for something that's trying to be a neuron, right? I mean, neuron needs an axon, it needs synapse organization, and these are just not showing up. Look at our downregulated genes, though, we now have overrepresentation of these terms. So as I wrap up today, I'd just like to mention that this isn't the only way that you can go about trying to attempt these things. There are other systems in place. They're often built upon DCAS9. I think the main drawback of this is that you don't have that ribonuclease activity, so it's harder to multiplex for your target loci. It's still possible, just a little bit more difficult. Some of them don't have the largest catalogs available. With this SunTag DCAS9, there's no dosage control, so you're expressing 5 effectors that are bound to this single chain variable fragment. There's no guarantee that you get all 5 represented simultaneously on your DCAS9. Whereas ours, they're directly fused, so all 4 effectors are going to show up. We're building a large, diverse effector catalog, and we're happy to expand it. with whatever you guys want. And the ribonuclease activity makes things really easy, and we're aiming for 10 times target multiplexing. So conclusions, just quickly. I think that this system has a great deal of potential. We've shown that we are able to use it to get some response, even with a very passive treatment, just removing repressive marks. But we anticipate that this would be greater if we, take advantage of the multiplexing and actively initiate transcription. And I just want to say tentatively, nothing definitive yet, but relative to normal neurons, we're seeing a bit of reduced post-mitotic character and maybe a bit more stem-like character. So big thanks to all the people that made this possible, and our emails are at the bottom if you want to contact